Thank you, everyone, for coming. I think we're going to get started here. First, I would like to just remind everyone that after the talk today, we're going to have a reception across the way in Basic Science Building, room 120, which is the Neuroscience and Cell Biology conference room. So it's my distinct pleasure today to introduce Dr. Sidney Brenner as the 2012 Mason Guest Distinguished Lecturer. Dr. Guest was a professor and chairman of the Department of Physiology here at UTMB for 22 years. And he made many seminal contributions to the field of blood coagulation. He, along with Dr. Barney Ware, demonstrated for the first time that thrombin was a proteolytic enzyme. He also did some of the seminal early work on an enzyme called urokinase, which is responsible for creating fibrin clots. At the time of his retirement in 1973, Dr. Guest was honored with the Ashbel Smith Professorship of Physiology here at UTMB, and served in this role until his death in 1991. In 1997, former friends, students, colleagues of Dr. Guest generously established an endowment to support his distinguished lectureship in his name. And now graduate student Serena Clark will introduce Dr. Brenner as the 2012 Mason Guest Lecturer.
Dr. Sidney Brenner, originally from Germantown, South Africa, received Bachelor of Medicine and Bachelor of Surgery degrees, as well as a Master of Science degree from the University of Witwatersrand in Cape Town, South Africa. Dr. Brenner then went on to receive his PhD from Oxford University under the direction of Nobel laureate Sir Cyril Norman Hinchelwood. Dr. Brenner arrived in England already thinking about DNA and became one of the first people to see the double helix model developed by Watson and Crick. After receiving his PhD, Dr. Brenner returned to South Africa briefly before accepting a position at Cambridge University. Along with Drs. Watson and Crick, Dr. Brenner became one of the founding fathers of the central dogma of molecular biology. Dr. Brenner theorized that an intermediate must exist between DNA and protein and confirmed the existence of mRNA experimentally. Dr. Brenner, along with Drs. Crick, Barnett, Watts-Tobin proved the triplet nature of the genetic code in a famous experiment exploiting proflavin-induced mutations of T4 bacteriophage. Further work on bacteriophage resulted in the development of the negative stain technique for determining virus structure by electron microscopy, which he quipped took the electron microscope out of the hands of the elite. While at Cambridge, Dr. Brenner had the insight to envision the simple soil nematode C. elegans as a model organism to elucidate the genetics of multicellular organisms. He recruited outstanding young researchers to help him on his quest, including his fellow 2002 Nobel laureates, Drs. Robert Horvitz and Dr. John Sulston, 2006 Nobel laureate Dr. Andrew Fire, and 2008 Nobel laureate Dr. Martin Chalfie, as well as our own Dr. Henry Epstein. C. elegans was an ideal model organism for many reasons, including the fact it is transparent, easy to grow in large quantities, is hermaphrodite, and is amiable to genetic manipulation and mutagenesis screens. C. elegans has become a model organism for many diverse fields such as development, aging, and apoptosis. For his seminal contributions in establishing C. elegans as a model organism, Dr. Brenner received the 2002 Nobel Prize Award along with Drs. Horvitz and Sulston. His work in C. elegans has ushered in the genomics era by pioneering new techniques for genome sequencing and assembly, the discovery of RNAi, and examining gene expression in vivo using GFP. Dr. Brenner currently holds several positions, including Senior Distinguished Fellow at the Salk Institute, Head of Molecular Genetics at the Okinawa Institute of Science and Technology, and Head of the Molecular Engineering Laboratory at the Singapore Agency for Science, Technology, and Research. It is my great honor to introduce to you today Dr. Sidney Brenner.
First, I'd like to thank you for inviting me here. This invitation has been delayed for my acceptance of it was immediate, but of course the arrival is what counts. And that's been delayed for several years. And of course I come here because Henry Epstein, organized it for me. He said I must come to Galveston. And of course I only had a very vague picture of what Galveston was like. And it's a much more interesting place. It's kind of, its Victorian environment is very interesting. And of course, I think that it's always good to come in and bring the message to lots of young people. So I'd like to talk now on the subject is reading the human genome. And let me tell you why I got to that subject. I discovered nobody knew what was in the human genome. Of course, everybody knew there was this particular gene and it did this, but nobody knew it in all of its glory, so to speak. Because the only things that have ever read it through are computers. And everybody knows that computers are incredibly stupid. They're very obedient. And what people have done is statistics on many of the genes, and there are plenty of papers on this. But what I want to remind you is the following. When we started the Human Genome Project, when a few of us began to talk about it in 1985, and we then likened it to sending a man to the moon. Actually, the amount of money was about the same. It was about $3 billion, about going to be $1 per base pair. But let me tell you that, of course, sending a man to the moon is easy. It's bringing him back that's the trouble. And I think the analogy is perfect, because the human genome is stranded on some metaphorical moon, and I think our big task now is to bring it back. Now, of course, we always knew, we always hoped that there would be left in it molecular fossils, that is, traces of the past. And I think that that was fully justified. So what I became very interested in is, could I reconstruct the past? And as you will see, I think it's a very interesting topic. Of course, it's hard to translate into any practical benefit, but I think a few of us should be allowed the opportunity to have a look into this. So, of course, many people have analyzed the human genome. There are endless papers written on it, but I want to just draw your attention to a number of things. What most people do when they do genomics, when they look at genomes, is they compare it to another genome. And they say, well, I'll compare it to the mouse, I'll count the number of differences, nucleotides, amino acids, and then I'll know the distance of these organisms from each other. And that resulted in concepts like the molecular clock, which I think is pretty well broken now. The clock doesn't really work. And also resulted in really a number of assumptions which have remained hidden in all of this. Now, of course, we know these genomes have been due to selection. They've been, part of them is selected, and so of course there will be traces of selective steps. But of course, one thing we want to really know about a genome is mutation drives it in different directions. And of course, the theory of natural selection says that some of these mutations are favorable, they will be selected for. What is uncertain in the minds of most people is what has become called neutral evolution. By definition, neutral evolution means there's no selective power attached. It's either because the nucleotide does exactly the same thing, or the amino acids resemble each other, and so the changes, it is said, will just measure the change. And Kimura made the assumption, which is widespread, that changes just due to neutral positions or neutral genes, they will only measure the mutation rate, because by definition you can't select for them. Now I'm going to show you that that's wrong, because what Kimura thought of was all the genes separate, like balls in an urn, or all separate things. And one of the important things, as you will see in a moment, is that genes are linked to each other. So it becomes plausible that we can see the indirect effects of selection because of linkage. I think if you link to a selected marker, then you get selected with a selected marker just because of linkage. There is no opportunity to segregate yourself. The first selection has to be for a whole genome. That's by definition. It's the results of a new organism. Then, if there is a particular gene that's been selected for, we have to whittle away the other genome either by segregation or by recombination until we emerge with this new definition. And everybody has said that neutral changes arrive in the organism simply by drift, by stochastic fluctuation. Now, I'm going to show that that is wrong. But before I do that, I want to tell you a little bit about the background. The surprising thing, and the one that still lacks a total explanation, is when you take the human genome and you measure the base composition, you will find is that, as Bernardi showed long ago before there was any sequencing, then it's constituted roughly of blocks about 300 base pairs of size that have a uniform composition, that is talking about A's and T's and G's and C's. But if you look at it, the composition ranges from extremely high GC to extremely high AT. And this fact that we are a mosaic of different compositional sequences has remained an enigma. Bernardi believed that it had something to do with temperature regulation, which is absurd, basically. But let me explain why he believed this. He found it true for all mammals. He found it true for birds, but frogs and fish didn't have this. Frogs, reptiles, fish. And so he said the isochors, as he called them, they were just regions which he separated by density in cesium chloride gradients. These were in fact correlated with the ability, because both birds and mammals can regulate their temperature. Okay, so he has this, but it's totally unclear to see why you should have DNA that varies, because you can regulate temperature, you might have thought, oh, you know, I have a high body temperature, so therefore I'll have DNA which is richer in G and C because it melts. It doesn't melt so. But this is nonsense as well. So it's hard to see what is the driving force to give you this mosaic interpretation. Most people have tried to show that the mosaic structures are correlated with various things, such as they're correlated with housekeeping functions. It turns out that a lot of the housekeeping functions, you know, like boring in things like glyceraldehyde, phosphate dehydrogenase, our genes was very high GC, actin, all of those. But let me tell you, you're not allowed to use this argument when you come to look at organisms that have germlines, okay? That have a germline in which the mutations occur and a somatic line in which the mutations are expressed. Okay? So you can't say that there's a different mutation rate for these genes because they're involved in housekeeping or expressed at high values, because you would have to test that where mutations are common. It would have to be true in the germline cells themselves, because you can't wait to say, oh dear, I made a mistake, wait until the organism is there. So selective forces in germlines work completely differently. And it's no use making kind of general molecular theories. So what I want to show you today is that the way we look at this problem, and the way to look at this problem is twofold. One is to have a very simple hypothesis, and that's easily tested. Now, of course, as you know, evolution is like cosmology. You can't start it from the beginning again. You can't run it again. So we must test it within the framework of what we can study. And as you will see, I think I can do this successfully. So the first question I want to answer is, given that nucleotides change, the base pairs change to other base pairs by mutation, how can I determine whether a genome, any genome, is at equilibrium? That is, you know, that as many C's change to T's as T's change back to C's, according to the mechanism. Because that's important, because that's the end, right? If the genome is at equilibrium, then by definition, it contains no information from the past. Okay, you can only tell that. Contain information from the past. You can only tell that if you can have a time-independent thing. At equilibrium, for example, could have been at equilibrium since last week or the year before, or for the last 200 million, or for the last billion years. We don't know, because equilibrium is just equilibrium. All right? So it becomes important. Now, in order then, the way I do it is I focus on certain mutations. And I focus on certain mutations only in the third position of the codon. Right? And I divide the codons of any organism into various classes. The codons I call 2-star. That is, there only can be one of two things. Okay? And you'll find that there's a whole family of codons like this, like phenylalanine, can only be UUC or UUU. And you can go through all of these and group them. Then what I do is, we just start with the 2-star codons, we ignore the rest. We take every gene and just reduce it to its 2-star codons. what we find is that there are two kinds of these. One in which we have the purines in the third position, like lysine is AAA and AAG, and one where we have the pyrimidines in the third position. But as you can see, they are mutations of the same base pair. It's a GC base pair going to an AT base pair. It depends whether the purine or the pyrimidine is on the crick or sense strand, or whether it's on the Watson or nonsense strand. But it's the same mutation. You couldn't tell the difference in terms of looking at it. So we think these two are very interesting comparisons. Right, so when you collect this all, you can ascribe to each gene a ratio. That is, for two small codons of the pyrimidine kind, we have this ratio of C to T. Right? We just measure that. We just count the number of codons. We pull them all together, count the number of codons like this, and then we have the similar ones on the purine side. So we get one number for each gene. Now, I have to tell you, if you don't know already, that computers do not like dividing by 0. So we therefore express this as the fraction of T. Okay? We then can take, for example, I can take all the bacteria that have been sequenced, and I'll just take a class of these bacteria. I'll take the class of gram-negative bacteria like E. coli, Serratia, and so on. And I can then express them just as a distribution. So you can bin all of these. You can say the first class, the highest GC, have a fraction of A from 0 to 0.04, 4%. And you go on through this until the highest you get is where the fraction of A is about 0.72. Sorry, I've just given you numbers. Of course, we do have things which are very high in A. Right, so we can do all of that, and then we can just take this and we can draw a map. Right, now how do we deal with this distribution? This is now a very simple process called the Markov process. And we do the following. Imagine then that the ratio of A to G and A fraction of A and G and so on is some number. And just for argument's sake, let us say it's half. It's 0.5, the average. That means the mutation rate is the same in both directions, just easier to think about. Doesn't matter if it's 0.3 or 0.7, the ratio, those just moves the mean. Now what you ask yourself is the following. Let's assume a kind of theoretical gene with 20 of such codons, right? So the average we'd expect there'd be 10 with C and 10 with T, right? Now what happens is we wait for the next mutation. And when that happens, the ratio changes. It'll become 11 to 9, right? Whether it's 11 of A and a 9 of G or the other way around doesn't matter, but the ratio departs, right? And now what happens is we ask ourselves, what happens if we wait long enough when there's a mutation in these codons happens again? Well, as you can see, the chances are that 11 to 9, you can bet it'll just go back again. But 9, 11's a pretty good probability. And so, in that case, it'll move down 1 again. Right? And in fact, you can calculate exactly this is, And technically, it is quite possible, even though the average is a half, to find things that are 100% G just by chance and 100% A. And you can draw this distribution very accurately just by calculation. Okay? Now you can ask yourself, I've got this distribution, and how do I discover whether the genome is at equilibrium? Well, you could ask yourself a rather general question, well, you know, does this distribution fit the theoretical? It doesn't fit the theoretical for one good reason, which I'll come to in a minute. But now we can do exactly the same with the purines, correct? And we find that they also will vary. And now what you can do is if you plot them against each other. Okay, so we plot for any subclass, what is the value of your pyrimidine codons? What is the value of your purine codons? If you do this, you can make a contour map of the genome. Okay, there are genes, it's just the family of genes that, and they cluster in circles. All right? So this over here are lots of genes, because this is the peak, it's the average, which have the correct ratio. And out on the outliers come the ones, because the two axes are, for the purines, that is, it is the A over R, and for the pyrimidines, it's you're plotting T over Y. Okay, it's just the total. Now, you notice if the genome's in equilibrium, what it's saying is that the migration of these triplets, here, the purine one, is independent of the migration of this, because this is just a random process. Right? And that means that the axes are orthogonal. They're at right angles to each other. Okay? And then no matter about the shape, you simply ask about symmetry. You can test this quite easily by taking something else which has no relation. For example, I plot the distribution of glycine codons, whether they have purines or pyrimidines. It's completely different. And there you find, then, if I plot it against, let us say, this, I get a set of ellipses, okay, with different spacing, because the mutation rate is different. But the axes are still at right angles. Furthermore, in some cases where there are codon abundance problems, okay, where actually you get many more for a certain codon, of one type, nonetheless, the axes are, so we have cases which you plot out, and what you find is that it looks something like this. Okay? And this is one where in one case it's completely biased to one of these, but nonetheless, the axes are orthogonal. That's a genomic equilibrium. If those two are correlated with each other, then the genome is in motion. Okay, it's a very important thing. So you can do this. You can also now see the difference between DNA transferred by DNA that's been transferred by horizontally, because on these maps you get little islands of a different composition, and you can tell whether the transferred DNA is at equilibrium. You can also see in some bacteria whether they've undergone a change, because you get the DNA at one place, and then you get a little causeway to run to a DNA of another thing. If you do Drosophila, you find that on this, and I'll show you in a moment what you find. You find that the Drosophila genome contains a lot of islands, which are way off from the bulk of the DNA. And if you look at what's in those islands, they're all the transposons. Okay, so this gives you a way of looking at the entire genome in a constructive way. All right? Now, so I can do the following, and I'm now just continuing with bacterial genome. If I do the yeast genome, these circles have a tail on them, like this. Okay? All right. In fact, yeast is not at equilibrium. Okay? There is this correlation between the two mutations. It's getting there, but it will still take some time. Okay? Because if you look over here you find a good old friend, glyceraldehyde phosphate dehydrogenase, which is both high GC and high, it's high G and high C codons. Okay? So we can tell that this, but Saccharomyces is at perfect equilibrium. Right? Now this is telling us this information from the past. Of course, you want to know, how do I know which way is it moving? Is all of this moving to higher GC, or is it moving down? I think you can look at the stacking of the contours, right? Because then they will follow the mutation rate over here. And this, of course, depends on the drag. So the contours are sometimes closely stacked, but sometimes they're very far apart. So these are just following. They are vertically delayed. That's what they are. They haven't yet got there to the equilibrium. Okay, now I want to tell you one other measure that you can do, which is a very interesting one. So I want to measure how far apart these genomes are. So let us take two genomes that are quite closely related, Serratio and E. coli. Actually, the Serratio's average is higher GC. Okay, so that's set by some slight differences in the process. And we can now ask ourselves the question, what is the distance between a Serratio and an E. coli genome? Now, what you can tell is the following. So suppose I take this genome here, the E. coli one which I showed you, and I take two of them, and let us say I have one set that is like this. Okay, and I have another set that is like this. This may be slightly extended. This may be slightly different mean variation. The genomes are quite close. But if I do this, then I can say, well, once upon a time, there was one genome. So let's see how strongly. So let's pick a particular gene. Now, according to us, if the gene started, so here's the mythical ancestor, okay, all right, and then you could argue the following. Let's suppose our gene started over here when this organism duplicated or gave rise to the separate line. And of course, I ask, where is it in E. coli? All right, and I might find it here. All right, and in Serratia, it might be over some other position. So of course, if I could compare this gene by gene and ask how many moves do they have, you see, because I can count each move of this is like going up. They can only move between these things by either going back or forward in the way that the mutation decreases. And so we could ask, if you had these, how far away from that have they diffused from each other? It's just diffusion that does it. And that turns out to be, quite a tricky thing to do. And so there's a much simpler way, which I want to show you because I use it a lot. Okay? So now I go to the other codons. These are the ones that have 4 positions. Okay, you can have either an A or a T or a C or a G. Okay, now what I look at is the following. I look at the ratio of C to G. That is, I take the gene, these are called 4-star, and I reduce it simply to a string of either G's or C's. You can also do the A's and T's, but I'll explain it from the G's or C's. And so for each gene, it's just got a pattern of G's and C's. All right? Now we look at what happens here. And the only mutations we're going to look at are called flips. Okay? A C and a G flipped. And the reason you do this is because the mutation rate is once again the same in both directions. If C goes to G, that is a CPG pair flips, then there's no reason why it shouldn't flip back at the same rate. It's a symmetrical thing. And all we do is we take the 2 genes. Now this needs very careful gene alignment. And we take the 2 genes and we ask, how many flips have you had? Right. And that gives us essentially a correlation of the 2 sequences. It turns out this process is quite slow, which we need this because we're wanting to traverse very long distances of time. So if I do this, and I do this for each of the genes of this, it's a very long process, I can just say, look, they're 96% correlated. Right? That means there's been only 2 changes. Each have had 2 changes. Okay, and you can work out the probability. It's quite nonetheless, it gives you a measure of the distance. So, for example, I find that Serratia is about 90% correlated with E. coli. Shigella is exactly the same as E. coli. I just think it's just another, from this purposes. So it turns out the correlation is 1. The genes are exactly the same. And this then gives you the thing. Now what I would love to do is to be able to put this measurement on real time, a real time base. And unfortunately I can't do that because I have no fossils. See, in this game, you've got an argument. So suppose you have a fish genome and you have a human genome. You can certainly measure them, and you can certainly say how far away they are from each other. But you can entertain 2 hypotheses. You can either say, well, long ago, the world was full of little humans, all right, and then they changed into fish. Long ago, the world was full of fish, and they changed into humans. Now, the only thing we've got that tells us which of these is wrong is fossil evidence. We do not have this for the microorganisms. So we've absolutely no idea. Furthermore, we only measure the integral over time. We measure the total number of changes over that time. And what we discover is, we don't know, it could have all happened, you know, on a sunny afternoon, and then after that everybody settled down and didn't change anymore. So we cannot get that resolution in time. All we can tell you is the rate. Okay? Now let's come and deal with the human genome, because this is what we really want to know. So we take the human genome and make a plot of it like this. Just take all the genes, just do the two-stop plot. And what we find is a measure, instead of finding a circle, we find a total correlation. All the genes that are high in C in those codons are also high. This has a little bit of structure, which is an abundant structure, but there's total correlation. So that says the human genome's in motion. Right? So let's draw this then just off of the distribution in one plane. And the distribution in one plane looks something like this. Okay, it's structured, and there is. This is a peak of about average 0.6, and this is an average of 2.22526. These numbers don't it looks bimodal. Okay, now, knowing that this is what the structure of the genome is, how are we going to interpret this model here? We can look in all these boundaries. What are the genes? Of course, have a look for glyceraldehyde phosphate dehydrogenase. It's over here. And actin is over here. And the opsins are over here. And if I look for very large, very complicated genes, they're all high 80. That's how dystrophin is over here. Okay. The other thing that's interesting, which you can plot here for the human genome, is what is the length of the gene? It's not the length of the coding sequence. That's usually very close. But the length of the gene is the size of the introns. Okay? So that increases through here. It increases at some value. The length increases as the abundance. So over here are small genes, small introns, very high GC, okay, and over here are things like dystrophin, which is the size of the E. coli genome. It's about 4 megabases, huge introns. Now if I go to the human genome and I say, what are these introns filled with? Well, they're filled with, mostly with alleles as well as other transposons. Okay, so I have these large, and if you can compare, as we have done, genes from, let us say, the pufferfish. See, the pufferfish is all over here. It's all over small genes, small introns, and so on. But let me say that's not a guarantee that that's the ancestral form, because we don't know which way this is moving. This is very hard to tell when you have a correlation. It could be moving backward, it could be moving sideways. Okay? All right, so now I have to tell you that that's what happens when I look at the 2-star codons. And so what I did very early on is I took a lot of genes, I lined them all, and I asked for this, and I didn't align them. I simply made a measurement of the 2 kinds of 2-star codons. All right? And I plotted for one of these. Actually, I can plot for the average of both, get a good number. I plotted the mouse genes against the human genes. They had to be the same genes. And all I plotted is this then. So, of course, the human plot comes out as theoretical, because binned in terms of, you know, where you start here, this is 0.04, and you're running to 0.72, and these are the different bins. So if I take the same ones from the mouse, and I plot them on this axis here, what I find is that I get a curve like this. Okay? I get a curve which crosses at the peak value. Over here, the mouse genes have a higher AT composition. Over here, they are lower. Okay? They are lower. So, you can't explain it by mutation rate. You've got to say these things have different properties. So what I thought was, oh, I know what's happening here. Okay, there's been a change, and in fact, everybody is converging to a uniform population if I waited long enough. And what I'll find is they're all at this crossover point. So if I wait long enough, I'll find that I'll have a uniform distribution. The mouse ones are moving towards this, so are the human. All right? And I can now say it's just a difference in rate. In other words, the mouse ones have got to start. But in fact, you can't fit a uniform rate to this at all, because you need very high rates at these very low GCs. There's no reversion. Okay, you can't get more GC than 100%. So from here you can estimate the forward rate of driving is about 20-fold faster. Okay, so this doesn't make sense. It says they're 2 different processes. Okay, and I'll tell you what the processes are. Right, so now what we have to do is start again and measure exactly the mutation rate. This we can do by taking the cow, for example, take its genes, and then when you find a difference, so this is now doing the 4th position, the 4-star codons. When I find that the mouse has a G, where the human has a C, I ask what does the cow have? Right? And therefore, if I know this, the likelihood is I can tell what was there originally, and I can tell which line had the mutation. Right? So I did that. I'm still doing it, because I want to collect very good data here, as you'll see in a moment. And then I realized there's a problem. Okay, and the problem makes life more complicated, but it actually provides the solution. Everybody knows that when you're in a CPG pair, okay, the mutation rate from CPG to TPG or CPA, which is the same thing on the other strand, is enhanced. Yeah. What it's due to, we don't have to ask. It's very likely due to some repair process, because These CPGs are usually methylated. Methyl C changes to thymine, and the repair process thinks it's a pretty good base. Okay, so one repair mechanism, one which excises thymine in base pairs, doesn't work there. Okay, it's only when it's unmethylated, it changes to uracil, and then it can see it as something novel and has machinery to excise these uracils and replace them eventually. So it would say that you get, looks like an enhanced mutation rate, but it's a lowered repair rate. And of course, that to us, that's the same thing. We have to take it into account. So if you do this, by the way, you can go through all of this with these things, and you can now ask the question, are all mutations with CPG pairs? Because I didn't do that with the coli, I didn't have to. Are all the mutations, perhaps they're all enhanced, perhaps the flips are enhanced. Okay, now, let's just look at the flipping business. So let's just see. So we have a set of codons that have no CPG within them. Okay, so they will be X, Y, and they'll be C or G, because we're just going to leave the A's and T's out. Now, I can make two classes of this depending on what's adjacent. So I can either have the next codon begin with the G, or what I call an other, that's C, A, or T. That's something to do with the sequence. Okay? So this will be, so the argument is, suppose flipping were to depend on this. All right, and let us suppose wherever you have a CPG pair, it'll flip faster. Correct? Then, in this class, there's no reason to expect it, because it'll just flip. But here, if we flip from a G to a C, we've generated a CPG, okay, which will just flip back. So the prediction is, these codons, if we measure the ratio of C to G, these should be half, just statistics on average, but these should be biased by some degree, right? So now let's look at the others. These have the following situation. They are X, C, or G, and then they can be C or G again. Or anything, that's the third position. Okay, because these are proline, alanine, and so on. Now, let us suppose I start with XCG. Well, I've got a CPG pair, so it'll flip. This won't flip, because this will change the codon. So this will change, and these will be biased to C. Okay? Because that won't flip back. Except in the case where the next codon begins with a G. Now, I know that this sounds very complicated, but it's the answer to everything, as you will see. Okay. So, you can do, so you first have to convince yourself. So the first thing I've done is I've done about 1,200 genes, all carefully aligned, and in which I can say for each gene, whether it's in the mouse or the human, I can tell you what the mutation is. So I can tell you, yes, there was a G here, it turned to an A. Okay, there was, you see, if it turns to something else, we just ignore it. We're only looking in the class. We don't care about it, we just eliminate it. So everything is done with these abstract strings. Okay, so now we can do something interesting. So we go and look, and the first thing we discover is that many more changes happen in the mouse than in the human. Okay? In fact, we can say for flipping, and the only flipping we do now is this subclass here. All the others are subject to these other conditions. So for this class of codon, the neutral flip here, and it turns out it is close to 0.5, okay? But the neutral flip here, and of course we change that, is the only one we measure when we compare. If one of these should have said, this is turned into a G, that's tough. It just gets moved to the other class and we don't look at it. Right? Eventually, you know, we're saying future universes, that'll come right again. Okay, so this gives you a very clean answer. And the clean answer is, on the average, the mouse is evolving 2.865 times faster than the human. There's roughly 3 changes in the mouse for every change in the human. Now, what does this mean? It's known that there were anomalies if you divided these things, or any changes, amino acids. Mice came out to be very young. You know, in other words, they couldn't work it out, how the mouse, because what's our evidence? So our evidence is the following. Rodents first appear in the fossil record, let's say 90 million years ago. It varies, 90, some say 110. Let's say 100 million years ago. Now, in that period, on the human perspective, when mice and men separated, okay, I had then a human gene which underwent a number of changes. But the mouse did 3 times more changes. So that means the only way I can make a consistency is the mice must be in the future. And indeed, mice are about 180 million years into the future. The human genome is very slowly evolving. Okay? So that's the first thing you do. Then you can make it. Then it becomes a bit more complicated. But this is very beautiful, this other thing. Okay, so what I do now is I have my plot. This is the landscape, high GC, high AT. Okay? Here, and it's this distribution with a human, something like this. I showed it to you, and it doesn't matter about that. Right. Now, what I plot on this is just this ratio for the flip rate. Okay? If I plot this one class of ratio, it's about 0.5. Okay. Now, if I plot these 2 classes together, because I can just say, I'll take the bias for this one is that these will be mostly G, the bias in this one, the other class are mostly C. All right, and I can take that and average them. Then if I plot that, that works out to be 0.8. Okay, it says one of them is 0.8, the other's 0.2. So they both give you 0.8. Okay, but you don't start that way. Okay, this keels over and is at the highest, you see it's about 0.6. Now this allows us to say you 4 times, the CPG will flip 4 times faster, okay, on average. But it comes to equilibrium here. So what does this mean? So here's the hypothesis, all right, and it's a cosmological hypothesis, you see? you know that if you do observations of the stars, okay, the further away the star is, the older the light is. The older the light is coming to you. And of course that old light undergoes what's called the redshift. So this is analogous. This is the GC shift. Right? It says that these are genes which haven't changed very much in their structure. Of course, their bases have fiddled around, but they haven't changed very much. So this is the original gene. Okay, this is it, a small gene with small introns, which is right. Okay, and now what has happened is that as this progresses, so this maps on time. This is one of those things that they would have loved to have had in thermodynamics, where the phase changes also reflected the time changes. But they can't do that, but that's what we'll say. Right? Now, so what we think has happened here is these are very old genes. This is the origin of the genome is over here, very high GC. And all the while they've been going through all these animals, they have progressed through this. The human lineage is here. And of course, every time an animal departs, it generates its own future. Okay, we can show that then the behavior of the mouse can be plotted there. But on this human time, the human genome time, this then is exactly what has happened, and round about here, these genes came into equilibrium. Okay? Now, one thing is interesting, you see? So these big genes over here are relatively modern. Okay, why? They've got big introns because they've simply undergone many more fixation changes. Right? Every time you put some more alus in, you have to fix that in all members of the species. So the fact that you get hardly any alu genes over here is not a measure they don't like to insert there. They'll probably be inserting all the time. You do not fix those changes. Okay, so there are two rates we have to consider. And over here we can count the number of fixation steps. The bigger the intron, the more it's undergone evolution. Right? And then we can look at a function, any function in the genome, and we can say today what it's about. So, I'll tell you, this is a very long story, and perhaps I've overdone it, but that is the story. And now it's got to be written up. But I think the big thing that emerges is we can reconstruct the past. I'd like to get a measure of the rate, the time it takes the human to pass through this, to the lineage to pass through this. I can't get it from other animals, because all genomes are of the same age, the present ones, the contemporary ones. I have to reconstruct the past as it existed, and that's a big job. So I think that we now have a picture that evolution drives genomes because it selects for various genes, and others come along. They come along just for the ride. They're hitchhikers, but they tell us, but linkage tells us what it's all about. Okay, thank you.
Sitting into consideration the comparison of RNA-coded sequences?
I haven't done RNA coding. I'm going to start to do that, but I can tell you that a lot of there are some of the microRNAs are old. we can tell that by looking at location. A lot of them are quite new, but we haven't done it systematically. You see, one of the problems with this game is because nobody makes proper sequence. They always want to do something and then write a paper and go on to the next thing. So we don't know if something's absent, there's really absent. Okay, so you have to validate all of these things. But we can do, you know, the fullness. So what's valuable in the genome is to have a perfect genome with no things missing. Because otherwise you can come to the wrong conclusion. And what I think is very clear is it's not also, I mean, it's interesting that not only are the coding sequence, the genes bigger, but the coding sequences are bigger. And they have been made by, you know, grafting various domains together. And so we can go through this and try to make this analysis. And of course, what's exciting about this is to try and see where are all the genes in the retina on this. You know, we can also tell quite a lot as to where they blanked out and where they arose. So that's the other job, is to do the genomes of the Cambrian explosion, which we will have to reconstruct from these genomes here.
The biochemist, you mean the G and C are more likely to be oxidized or demethylated, I'm sorry, deaminated, and they are more likely to produce GC to AD. Converting AD to GC is much more, not so easy to visualize in terms of replication.
Yeah, I think, you see, this is, I mean, but you see, thank you for your, but it's a completely irrelevant, because I've got to tell this from the existing genome. What you've done, what you're telling me is what has been found as experimentally. I've got to prove that those conditions existed. Okay, now I can do certain things like this by arguing about repair mechanism. And for example, we know how many genes, what is the speed of evolution of genes that are unpaired. So it's a very funny business. Then of course when you get a transposon, it's a heterozygote, you see, and now it's unpaired. So we can look at genes that are unpaired. There's a small family of genes on the Y chromosome that we can look at. The speed of evolution is much faster with these, and I think that's because there's no recombination or repair. There's nothing to pair up. From this we must deduce that you know, for a long time these transposons exist in a genome in single copy, so you can't take any measurement, because until they come under the regime of being repaired by recombination, they won't stabilize the thing. So people have tried to look at transposons, made arguments about this. We need to know all these other things. So I think this is interesting, but again, you have to deduce it from this, and it's one of the problems if you talk about it in this way. Now, trying to work out how the rods and cones in the eye did this. But of course, I've taken it back to a time where it's nonsensical to talk about rods and cones. We probably should be talking about cods and roans, you know. We just don't know what they are.
The other question is whether CPG primarily present and methylated primarily in the regulatory sequence that is in the mammals. You know?
Yes, all I can do is this. I can tell you, I can tell you what exists today, right? But I can tell you that there is a bias for CPG that existed a long time ago, and it's left the mark. Okay? So it's left the mark, there's no doubt about this. And that it started wherever this date was, which I still can't tell you, has continued because it's come into equilibrium. So that then is the way to look at all of these things.
Yes.
When you're comparing the rate of evolution between different organisms, how do you correct for the rate of the generation time?
I don't do it.
Mouse, for example.
No. It could be the main reason. All I'm telling you is that, you see, this is a very strict, a game with very strict rules. You've just got the two genomes, and everything you deduce has to be from the sequence. Okay? And I don't know, it could be that the mice do all of this. And you see, from the point of view of mice, we are exceedingly primitive. We are way back, on the mouse scale. So it's just that organisms move at different rates, they have different repair mechanisms, but we can only deduce that from the genome. Okay, that's the rules of the game. You're not allowed to bring in, for example, people say, well, for example, transposons could be hopping around in the brains of everybody like crazy. But if they're not recorded in the genome, it's of no consequence. You see, it's of no consequence to an evolutionary argument.
Okay, great. Thank you very much.
Okay.